I'm going to go through the review really fast, so because I do want to start muscle today. So we will hopefully start muscle, but if we don't, then we don't. Everyone have their review sheet on you? Who turned the lights off? Whatever. So, whoever wants to record, have you started recording?
Yes.
Okay. All right. So this list is not comprehensive, which means you come to me with, oh, this is not on your list, or you never taught me this. Will be met with a lot of love because you're all recording. So infer at will. All right. So well, it's pretty comprehensive. Okay. So I think I'd be surprised if there's something on the exam that's not on this list. Okay. So the exam is next week. So Tuesday class, you'll be taking it on Tuesday, and Thursday section you'll be taking it on Thursday. Lab is really light. You're either wrapping up whatever you did with the sensory. So lab is very optional next week. Okay, so if, but remember, you take your lecture exam in lab. So I have to be really careful how I announce things. So you do need to show up and take the exam. But once you're done with the exam, you're welcome to go home. There's really not much planned to do. I will update you on the announcements. I have to look at the overall schedule. If I feel like it's really, really light, we might do an electromyogram, which is perfect because we're doing muscle, right? So we might do an electromyogram, which is not that long. Okay, it won't take you that long. It's not like EEGs or something. It's a little bit easier than EEGs, so we most likely will do an electromyogram. I'll update you. Okay, let's see how it goes. On to the review. So body organization, this is where cells make up tissues, tissues make up organs, organs make up organ systems. Please note that in an organ system, the organs don't have to be connected. So example, the excretory system, remember, has liver and the lungs and the kidneys, and none of them are connected to each other, correct?
Yes.
So in an organ system, the organs do not have to be physically connected to each other. Then we went into mechanisms. Mechanism is a step-by-step explanation of how a process occurs. Mechanism is probably one of the most important words in this class, so please make sure you understand it well. A linear effect or a linear mechanism is where A affects B, right? For example, insulin will cause blood sugar to be taken up. If that's all that's given, right, that's not a feedback mechanism. Feedback means you're coming back and giving me input. Do you understand? There has to be a coming back portion there, right? So, for example, let's say there's injury and immune cells are gathering around that injury site. Yes? That will be an example of a linear effect. Are we clear? Because I haven't really given you any feedback there, right? It's not a feedback mechanism. Are we clear about this? Yes. You will need to know how to distinguish a feedback from a linear from a cycle. Okay? A cycle is also not a feedback. Okay? A cycle could simply be like the Krebs cycle, where you're going around, right? And round and round. There's no feeding back, right? So it's like you submitting your your paper to me and me giving you back your paper, but with nothing on it. Makes sense? Just a grade, right? There's no feedback there. Right? Because I'm not communicating back to you. Am I making sense? I'm just giving you back your paper. Right? So these were all very subtle points, but they're really important for later. Feed forward is a mechanism that we're really not going to go into much, but feed forward is what the body does in anticipation of a certain stimulus. So feed forward has to do with anticipation. Anticipation. For example, your insulin levels begin to rise, right, when you see a bun cake, right?
Yeah.
That's a feed forward mechanism. Okay, that's an example of a feed forward mechanism. In anticipation of a highway exit, your body begins getting ready to turn, right? That's a feed forward mechanism, right? So you need to know how to tell all of these apart. Please take notes if you can, but I am going to be speaking really fast, so you may not be able to write things down. The way you should use this review is if you don't recall a term. You're like, wait a minute, I don't know that term. So wait till I get to that term and then take notes. Or nothing else, just circle that term so you can go home and review. This is all in your notes that I've given you. I've given you the notes. They're all in the notes. Okay? So feedback mechanism. This is what maintains homeostasis, and particularly negative feedback. So when we say the word homeostasis, you should immediately think negative feedback, not positive feedback. Positive feedback is not homeostasis. Whenever I say something as not, I would note that down because that's actually really important. So positive feedback cannot be homeostasis. Negative feedback and positive feedback, let's define them first. But before we do that, let's talk about what feedback actually means. Feedback means A goes to B, goes to C, and C affects A or B. Okay? So you go backwards, you go back to where you started, right? So that's feedback, and you're affecting them, right? So the components of any feedback mechanism, very, very important. And this week in lab, we did reflexes. They have the exact same components as a feedback components. Okay, same thing. You need a sensor, something that is sensing the variable, and then you have an afferent pathway, something that takes messages from the sensor to the processing center, and from the processing center, you have efferent pathways that take decisions back to the effectors, and effectors will affect a change. Affect meaning will produce a change that will, be a change, whatever be that change, right? Now, if the change is opposing the initial stimulus, that would be a negative feedback. And if the response is augmenting or enhancing the original stimulus, right? That would be a positive feedback. So again, a very common mistake is people think increase means positive feedback, decrease means negative feedback. That does not work. Okay, that is not true. That is not at all true. And I will purposely give you examples where it goes the other way. Okay, so please learn your definitions properly. Negative feedback is where the response and the stimulus are opposites. Positive feedback is where the response and the stimulus are the same. Okay, very simple. Negative feedback, N for negative feedback, N for normalcy. Negative feedback is the only thing that controls homeostasis. Positive feedback does not do homeostasis. Positive feedback is injury. Positive feedback is chronic. It is very unusual and rare. Negative feedback is the most common, and it's 24/7. It's constitutive. It's always on. Negative feedback is always on. Yeah.
Could you give an example of how an injury can be a positive feedback?
So positive feedback is when the platelets gather, right? So there is injury. Okay, blood slows down, and platelets begin to gather at that site, right? When one platelet gets there, it releases factors that will attract 100 platelets there. Now you have 101 platelets. They will release factors, bring 1,001 platelets, on and on and on until the injury is sealed. Okay, until you plug it. Okay, so that's positive feedback. You should know examples for these. Okay, blood sugar regulation is negative feedback. Injuries, childbirth. Okay, these are positive feedback. Here is another positive feedback mechanism. In an action potential, immediately your brain should go to the graph. Are you recollecting the action potential graph? In an action potential, going from rest, start visualizing, going from rest to threshold. Going from rest to threshold. Action potential is 35 points on your exam. Okay? I know this because I wrote it. So 35 points on your exam is action potential. You better know that code. Okay? So, again, are you visualizing the depolarization to threshold? Right? Is that slow or fast? Okay, everybody imagining the action potential graph from rest to threshold. Is that a slow or a fast depolarization? Slow, okay? It's a slow depolarization, and that's an example of positive feedback. Okay? Slow depolarization is positive feedback. Fast depolarization is not. Fast depolarization is not an example of positive feedback. How? Because when one sodium goes in, it starts repelling the M gates. So M gate starts flickering. That causes another 2 or 3 sodiums to go in. Now you have 4 sodiums. That's going to flicker the gate even more. As the gate flickers more, more sodium gets in. More. Do you see this? Yes. Until you hit Tmax. Are we clear?
Yes.
Which would be threshold. Now, thermoregulation. Thermoregulation, when it's a controlled thermoregulation, like fever, example is a fever cycle, that would be a negative feedback, right? So what happens in a fever? Your body has infection, and the immune cells detect that infection. They release chemicals that will set the temperature in the hypothalamus to be higher. That's very easy to do. They basically bring down the proteins that are responsible, okay, for signals to cause you to, that basically shut down the cold response, right? So if the thermostat is set to 100 now, instead of 97, and you're at 99, your body now perceives itself to be cold. So it'll warm up, right? And then you warm up. When you warm up, let's say you go to 101. Well, now your body wants to cool itself down. So this is why during fever, you go through fever and chills, and fever and chills, and fever and chills. Are we clear?
Yes.
There are cycles. It's classic negative feedback. The hypothalamus is involved, big time. So in body temperature regulation, if hypothalamus is involved, it's negative feedback, for sure. And fever cycles are negative feedback. You can always look this up on AI or look this up on YouTube and watch animations where you see how this happens. And you can also look up on Google Scholar exactly how immune chemicals reset the thermostat, the hypothalamic thermostat. It's actually very straightforward. You can go read it up on your own. It's again, downregulation of certain proteins in the hypothalamus that gets this done. Okay? We don't need to know that. I'm not covering all that detail in this class. Hypo and hyperthermia, which are long-term signals. And again, I really need you to know this for the class. I may not test you on this exam, or I may, but it's very, very important in physiology that if a stimulus is long-term, okay, it is stupid of the body to keep on responding. Are we clear? Yes. If my blood sugar is going to be high all the time, right, or your blood sugar is going to be high all the time, the pancreas will stop responding because that's dumb. You're wasting energy, right? You're just going to reset your set point. All of a sudden, a blood sugar of 150 is now normal. Make sense? Yes. So when your blood sugar goes to 100, you actually get hypoglycemia. When 100 is supposed to be normal for any non-diabetic. Are we clear? Yes. So long, persistent stimuli will reset the set point. Write this down. Very, very important. You're going to see this every single chapter. You're going to see this. Okay? Persistent signals will reset the set point. Okay? And that's what happens with high blood pressure. That's what happens with high blood sugar. Okay? And high body temperature. And that's what we talk about in hypo and hyperthermia. So So this is another really important point. Long-term stimuli will shut down negative feedback mechanisms. Long-term stimuli will shut down negative feedback mechanisms or reset the set point. But if it's going to be super persistent and extreme, then eventually negative feedback mechanisms get shut down, like in the case of hypo and hyperthermia. So if your body is always cold, and when I say cold, I'm talking pretty severe, right? Your body temperature is 97, right? Let's say that your temperature is always 89 degrees, right? That's really cold. And that can happen when people live in extreme cold conditions, but most likely it happens in disease states, okay? There's something called Raynaud's disease, and their fingers look frozen. I'm not kidding, they look white and frozen, okay? And so that's one disease. Also happens with hypothyroid patients, extreme hypothyroidism, where you don't have enough thyroid hormone. And if thyroid hormone's not there, your heart doesn't beat as fast, you don't get blood circulation. Blood circulation does not go to a particular organ, it's going to feel really, really cold. Are we clear? Mm-hmm. So if you have persistently extreme cold, that's hypothermia. There, what happens is the hypothalamus stops responding. Okay? I had a student write once when we used to have essay questions, the hypothalamus dies. Hypothalamus doesn't die. Okay? It just stops responding. Are we clear? Mm-hmm. If hypothalamus dies, you can't be alive. Does that make sense? Yes. Don't make things up. So hypothalamus stops responding. Okay? It's like somebody who's constantly arguing with you. Okay? Mother-in-law. You just learn to ignore them. And that's what hypothalamus does. Okay, so then what happens? Your body temperature, okay, goes down. Your heart rate immediately starts going down. Your heart can sense blood temperature. So when blood temperature gets cooler, your heart starts slowing down. When heart slows down, your metabolism slows down. When metabolism slows down, your body temperature goes down even further. Because only when you metabolize, you burn calories and you get ATP, right? So it's going to be a spiraling down, right? You started with low temperatures, now you end up with even lower temperatures. Positive feedback. Okay, so that's hypothermia. Hyperthermia, again, all this detailed in your notes. Take a look at your notes. Hyperthermia, okay, is when the outside temperature is super hot, right? And very easy, go to South America, go to Brazil during summer, right? And if you don't take proper precautions, your body will get really hot. And when your body gets very hot for several hours, hypothalamus shuts down. It's not going to respond. And then your heart starts racing. And then you start sweating, right? And then at one point, the brain says, that's it. When your heart rate goes way up, it puts that parasympathetic brake. And people faint. That's called a sunstroke. Does that make sense? And we did parasympathetic, so you know that, right? So right when class was over last time on Tuesday, there was a question about why do, when people get scared, why do they pee and poo their pants, right? And also, why do people faint? Those are all parasympathetic overriding the sympathetic system to protect your heart. Are we clear? Mm-hmm. Otherwise the heart would explode. Any questions on that block? I'll give you 30 seconds to circle what you don't know. Yes?
If the hypothalamus stops responding, can it respond again?
Yes. Once you have a sunstroke, don't you come back to life?
Yeah.
Yes, so hypothalamus comes back, yeah. But that really depends on the stimulus. And for a diabetic, for their blood glucose, even if the blood glucose is super normal, for me, this whole year, it's been wonderful, right? But no, it's still, for me, anything below 150, I start shaking. So even though so it takes a long time, it takes 1.5 years for that to set back to the normal sugar. So some parameters go back very quickly, some parameters go back very slowly, some parameters don't go back at all, like blood pressure. as far as I know. It almost never goes back, particularly if you've been hypertensive for at least 5 years or more. So that's why people are screaming, take care of your blood pressure. Okay. Any other questions? Okay, cell membrane composition, phospholipids and membrane proteins, right? It's a mix of phospholipids and membrane proteins. It also contains cholesterol. The more cholesterol you have, the more rigid the membrane becomes. The more cholesterol you have there, the more rigid the membrane becomes. Okay? And understand that this is not just dietary cholesterol. Okay? There are people whose cholesterol, blood levels of cholesterol are really abnormal, right? And yet, their membranes are not so rigid. Okay? There are people with hypercholesterolemia who exercise a lot and their membranes are not so rigid. Does that make sense? So it's not so straightforward. But for the purposes of this class, we'll keep it simple and say you got more cholesterol in your membrane, the more rigid it's going to be. That part is inarguably true. Okay, no question about that. The more cholesterol you store in your membrane, the more rigid the membrane is going to become. Clear? And we know that exercise makes your membrane less rigid. That part is proven. Okay, exercise will make your, particularly muscle membrane, it makes muscle membrane more fluid. Okay, even something simple like walking 30 minutes a day. So the fluid mosaic model basically describes your membrane as flexible. So the word fluid means flexible. It can easily fold over, FFF, right? So it can easily fold over. And that's important for insulin response and many other hormones, but mainly insulin response. Okay? And in fat cells, the folding over is very easy because fat cells have so much lipid in them that the membrane is always very supple, right? So if insulin is not working on muscle, or if it's not working on the liver, which it does when you're exercising, so if you're going to be a couch potato, it's going to go straight to the fat cells. And in fat cells, insulin is lipogenic. It generates more lipids. Okay? That was one of my rotation projects in my PhD first year. A very interesting project, actually. So, and insulin causes adipocytes to differentiate big time. So insulin has a very powerful response there. In terms of the mosaic, so what does mosaic mean? It refers to membrane proteins that look like tiles in a kitchen floor, okay, or a bathroom floor, or a library floor. Okay, so tiled appearance would be membrane proteins. Peripheral membrane proteins are loosely attached to either the membrane or to integral membrane proteins. Okay, so peripheral membrane proteins can be attached by themselves to the membrane, one side, or they can be attached to integral membrane proteins, okay, which cross the membrane fully. Everybody visualize this? Integral membrane protein is a big bridge that goes through both layers, both phospholipid layers. And peripheral membrane proteins only attach to one side. And they purposely attach very loosely because they need to dissociate for signal transduction to happen. Very, very important point. You're going to need this as soon as we hit the endocrine chapter. You're going to need to know this. Peripheral membrane proteins are signal transduction proteins for the most part. Peripheral membrane proteins have three main functions. They're signal transduction. Okay? They can serve as cell-cell junctions. Cell-cell junction is on this exam. You need to know cell-cell junctions for this entire class. Okay? So cell-cell junctions are proteins that connect cells together. Okay? And I really hope you went over this in anatomy. Have people heard of cell-cell junctions? Okay? So you got tight junctions. Okay? Tight junctions, you need tight junctions if you want two separate environments. If you want to separate the environments, you want tight junctions. Tight junctions do not add strength to a tissue. It's very important to understand that because the word tight implies that it's adding strength. It's not. Tight junctions do not affect strength. Are we clear? Yes. Okay, I will ask it. Tight junctions do not affect strength. Okay? The second type of junctions are desmosomes. Desmo means bridge, soma means body, and desmosomes, what desmosomes do, they add a lot of strength to the tissue. So the more desmosomes you have, example, your skin, your epidermis, and your heart, cardiac muscle. Cardiac muscle has desmosomes. And remember, your heart needs that strength because the heart pumps several liters of blood a day, right? So it needs the strength against tearing. And that's called tensile strength. Tensile strength means the ability to resist tearing. Okay? Somebody thought in my anatomy class that when I say resist tearing, it's resist tearing. And they said, why would tissues cry? And I'm like, no, sorry, it's the same spelling, but totally different pronunciation. You're resisting tearing. Yes? Tearing. Okay. So, integral, and the third type of junction, really important, gap junctions. So, tight junctions, desmosomes, and gap junctions. Gap junctions. Gap junctions are hollow. They connect cell cytoplasms together. When you have gap junctions in a tissue, you're going to synchronize their activity. Think about it. If you're connecting two or more cells together, that means when I turn one on, the other one is also turning on, correct?
Yes.
Okay, so gap junctions ensure synchrony in tissues. Synchrony in tissues. So those are the three junctions. Okay, tight junctions, gap junctions, and desmosomes. And that's the cell-cell junctions part there. Okay, and what is signal transduction? Signal transduction is when you have a serial activation of proteins. Serial activation of proteins. It's just like running a relay race. I pass the stick to you, you pass the stick to the next person, that person passes the stick to the next person, and so on. So signal transduction is serial activation of proteins inside a cell. Serial activation of proteins inside the cell. You're going to see this up the wazoo when we do the endocrine system. Okay? You already saw this in the nervous system. You already saw this in the nervous system. Direct or indirect acting, that's signal transduction.
Indirect.
Indirect. Okay, good. Indirect acting neurotransmitters, they do signal transduction, right? They have to activate several proteins before they turn on some ion channel. Okay? Which means indirect acting is always going to be
Slow.
slow. Okay, or slower than direct acting. Okay? I did all these onto molecular, before we go to molecular transport, integral membrane proteins. What do? They cross the entire cell membrane. They cross the entire cell membrane. Okay, they cross the cell membrane. And so they're used for several things, right? They provide stability to the membrane. That's why they're called integral, meaning if you remove them, the membrane collapses. Okay, so you need to understand that. So I could ask you a question like, if you use a very special type of detergent that removed all the IMPs, okay, what would happen? The membrane would simply collapse. Are we clear? The cell would cease to exist. Okay. The integral membrane proteins provide stability. They're cell transport proteins. Cell transport proteins. They're hormone receptors. Okay. They help peripheral membrane proteins attach, right? So they're attachment site for peripheral membrane proteins. Again, all this is in your notes. So if you have your notes in front of you, you can add to it. Okay, so this is all in your notes that I've given you. Okay, and then integral membrane proteins can also be enzymes. They can also be enzymes. They're part of cell-cell junctions. They are part of cell-cell junctions. And lastly, they can be a molecular billboard. They can display antigens for the immune system. So they're important for cell recognition, right? So immune cells recognize cells as self or non-self using integral membrane proteins. Are we clear? Are you recalling all this? Yes? Okay. On to molecular transport. Molecular transport is when you transport a finite number of molecules at a time. Finite number of molecules at a time. And bulk transport is when you transport a large molecule or even an organism, or you transport in bulk. Remember the words bulky and in bulk, right? Bulky means one molecule, one big protein, or bulky or in bulk means a whole lot of glucose molecules. That by individually they're tiny, but when you transport 1,000,000 of them, that's going to be bulk transport. Bulk transport requires vesicles. For vesicles to work, you need cytoskeleton. For cytoskeleton to work, you need calcium and you need energy. Do you see how all this connects? Yes. Those are your requirements for bulk transport. For bulk transport, you need vesicles. For vesicles to work, you need cytoskeleton. For cytoskeleton to work, you need calcium ions and energy. Okay? And cytoskeleton, right? So cytoskeleton to turn on, you need calcium ions and energy. Are we clear? So, and if you can imagine this, you're laying the rail tracks as the train is moving. Are we clear? So it's really amazing. It's like a sci-fi movie. You're laying the track as the train's moving. Okay? You're going to need energy for that. Clear?
Yes.
Okay. So bulk transport is of two types. You got exocytosis, which is called secretion. Exocytosis is more commonly called secretion. That's why secretion requires calcium. Secretion requires calcium. And endocytosis is when you take things in bulk or bulky, right? So endocytosis, if you're taking solids in, it's called phagocytosis. If you're taking liquids, it's mostly water, okay? And that's called penocytosis. And third, you have special molecules like cholesterol. When you take those into cells, that's called receptor-mediated endocytosis. In fact, HIV infection happens that way. It's through receptor-mediated endocytosis. Okay, again, if you're not recognizing those terms, ask or put a circle and make sure you review it at home. I don't see any of you during my office hours. Again, I want to reiterate that my office hours are every day from 8 to 9 on Zoom. Okay, 8 to 9 on Zoom. Please feel free to come. If you want to just sit there and study the whole hour, feel free, okay? You can do whatever you want. Sit there, study, or sit there and chat with me. No one comes, so it's not like a big deal. But usually I have a bunch of 103As show up. So, and they're all worried all the time, right? Your class is a lot more chill. Well, maybe because I haven't seen you yet. But feel free to come. I'm there. I'm there tonight. I'm there tomorrow night, okay? So I'm there all 5 weekdays if you have questions. Okay. Let's see, where are we? So bulk transport's done. Molecular transport is classified into two. You've got passive transport or active transport. Passive or active. Passive means energy is given off. Active means energy is required. They're exact opposites of each other. Okay? Makes sense because passive, what are you doing? Along or against?
Along.
Along the gradient or down a gradient. And a gradient means difference, correct?
Mm-hmm.
True or false? Concentration is the only gradient that exists.
False.
False. You should know voltage gradient exists. You should also know pressure gradients exist. Pressure. Okay, that's how we transport gases, because you have a pressure gradient, right?
Yes.
So all of these exist, so keep that in mind. Molecular transport requires gradients. It requires gradients. So if I give you a question that says, oh, a particular transport has absolutely no bearing on what the gradients are or what the concentration is, 100% that's bulk transport. Are we clear? Remember, the Costco truck doesn't care how many pasta packets you have in your garage. Make sense? If you have only one pasta packet left at home, then you're going to go to Safeway or wherever you go and go get another or order one from Amazon. Are we clear? You're going to do that, but Costco truck's not going to care how many packets you have or don't have at home. So the same way, bulk transport does not care about gradients. Please, for heaven's sake, draw that entire chart out at least once or twice. Cell transport is 50 points out of 100 on this exam. It's the biggest chunk, of course. Okay, it's a big deal. So I better have simple diffusion. Oh, there you go. I was going to say. Simple diffusion does not require any proteins. So here's another exam question. If I use a very special soap and I remove integral membrane proteins, so listen to my question carefully. If I used very special soap and I removed integral membrane proteins, yes? Mm-hmm. Which of the following can still enter the cell? Okay, A, glucose, B, sodium, C, potassium, D, carbon dioxide, and E, none of the above. How many people think D, carbon dioxide? Okay, answer is none of the above. If I remove IMPs, will the cell exist? Then is transport even a point?
No.
This is how you need to think. So this is the difference between blind memorizing and critical application. And this was a TEAS question 3 years ago. I took this out of a TEAS exam. Okay? Because one of my students came back and said, this question really bothered me. And I said, wow, they asked this? And he goes, yeah, it was there. And he remembered all the choices. So think. I just told you, if you removed all the IMPs, the cell would collapse. When the cell collapses, who cares what can enter, what can't enter? Nothing can enter. It doesn't exist anymore. Right? However, if I say, right, you're creating an artificial membrane with no proteins in it. Now listen to my question. I'm creating an artificial membrane. Which of these molecules can diffuse across? And the answer would be water, urea, steroids, okay, cholesterol, correct? And gases, right? Oxygen, carbon dioxide. Are we clear?
Mm-hmm.
So simple diffusion happens without the help of any protein. So read the question carefully and think. That's why I'm giving you extra time. You have an hour and a half to do the exam. Everybody finished in 40 minutes. What's the rush? Which mother-in-law are you running out to see? That's not good. If you end up getting a 98 and you finish the exam in 10 minutes, I'm going to shut the F up. I'm not going to say anything. I have nothing to say, right? But if you're going to get a 40 on the exam and you finish in 40 minutes, I'm going to yell at you. Why did you finish so early? Hello, good morning. That's logical, correct?
Yes.
So gauge for yourself, right? And look at the exam that you took just now and say, did I rush through this? And then gauge for yourself. You don't have to answer me, you have to answer to yourself so that on the next exam you don't pull that stuff. Spend some time. My questions are not like on Jeopardy where you just vomit the answer. Okay, you need to think. Even my anatomy exams are like that. Okay, you do need to think. Okay, all right, back to this. So gradients are important for simple diffusion. So things keep on moving until the gradients are equalized. Okay, then you have facilitated diffusion. Polar substances like ions, okay, and glucose and amino acids. And yes, you need to know these. Glucose, amino acids, ions, okay. Those cannot cross the membrane because of the hydrophobic barrier, right? And we call this selective permeability. Selective permeability. If that's not up there, make sure you write it down. Selective permeability. That's why I said this is not a comprehensive list. So those molecules will move through the central hydrophilic tunnel in these IMPs, correct? Remember the hydrophilic tunnel, right? In the IMPs. So things move through there, okay? So those could be carriers or channels. But tell me, both carriers and channels have to be blank. What? No, carriers are active transport. Good guess though. Listen to my question carefully, process and then answer. Both carriers and channels have to be blank. Conditionally. That's too generic. But you're on the right track. Channels don't have shape change. IMPs, right? They have to be integral membrane proteins. Hello, right? If they're halfway, can you transport? If the San Mateo Bridge is only half built, can you cross?
No.
Think, right? Of course, you have multiple choice, right? You'll have all the answers there, but you need to think. Okay. So yes, all protein transporters have to be IMPs. Now do you see why I took 15 minutes and taught you IMP functions? Connect them together. If you're not going to spend time on this, it's going to show on the exam. If you're just going to sit there for an hour and a half and just stare at the notes and then have Facebook open on the side or Insta, whatever you young generation does, to distract you, and you're not 100% into these notes, you're not going to do well. No matter how easy I make the exam. Because it's not my exam that's tough, it's a concept that's tough. Yes? Mm-hmm. So please make sure that you're thinking through all of this and spend some time on it. So that's facilitated diffusion. Facilitated diffusion can be channel or a carrier. It can be both. The difference between channel and carrier is the degree of shape change. Carriers have to undergo severe shape change, okay? And channels undergo minimum shape change. If you remember one, you remember the other, okay? So channels can only be, for each of this is a question. Channels can only be facilitated diffusion. Channels can never be active transport. Are we clear?
Yes.
Okay. Whereas carriers can be both. They can be active transport or passive transport. And carriers can be uniport. Can channels be uniport?
No.
That doesn't make any sense, okay? So remember, carriers are uniport. Carriers can be co-transport. And co-transport can be symport or antiport. Symport, two substances, same direction. Okay? And antiport, two substances, opposite direction. This will come bite you big time in the kidneys. When we do the kidneys, you need to know the code. Okay? Sodium, potassium, ATPase, you should know step by step how this works, right? So it's a cycle. Sodium binds, 3 sodium ions bind, that will attract ATP because sodium is positively charged. ATP has a charge of -9, so that's going to get attracted to sodium, right? ATP gets hydrolyzed. Okay? You put a phosphate on the pump, right? The pump will do what? Flips. The pump inverts, literally. Flips. Okay? And then sodium is let loose, right? And then potassium's bind, right? Mm-hmm. And then the phosphate is removed, correct? Mm-hmm. And that causes the pump to flip back. And that's how potassium comes back. It's a very simplified view of things, okay? That is not at all how this works, okay? It's like cartoon. It's a Tom and Jerry version of what truly happens, okay? So, but sodium, potassium, ATPase, 3 sodium ions and 2 potassium ions in opposite directions, so it's an antiport, per ATP. Okay? You need to know those numbers. Anything that involves a protein, anything, it's not just transport, anything that involves a protein will have a Tmax, which means transport maximum rate. True or false? Transport maximum rate means 0.
False.
False. Transport maximum rate does not mean 0. It means a high rate that doesn't change. Are we clear?
Yes.
Okay. So everything has a Tmax. Okay. Especially if you require protein to do the job. Because proteins are specific and saturable. Specific and saturable. So when I say chemical specificity, that means glucose channel will only bind glucose. That's chemical specificity. Are we clear? Mm-hmm. Saturability means you hit a Tmax. You will hit a maximum rate. Okay. With this, all done. Okay. So you should know all the steps to active transport. Active transport, primary active transport, transports molecules and utilizes ATP. I want to be careful here because in 102 you studied mitochondria. They don't use ATP, they produce ATP, right? But that's still active transport. Are we clear? So you can do active transport without ATP. There are other sources of energy. Are we clear?
Yes.
So I just want you to know that a primary active transport uses energy and transports all in one. It's all in one. I have a part of me that uses energy and I have a part of me that transports. Okay? That would be primary active transport. Primary active transport can be symport, can be uniport, can be antiport. Are we clear? There are examples of all of them. Make sense?
Yes.
Okay. Now, secondary active transport is very specialized. So primary active transport creates gradients. I don't like creates. So I'm going to say primary active transport establishes gradients, whereas secondary active transport does what? Uses those gradients. Okay? Secondary active transport has to use one gradient to push a molecule against its gradient. So sodium coming in from the outside will release energy only if sodium is low inside, for which primary has to work. Hello, do you see all these steps? So when sodium comes in, gives off energy. That energy is used to push glucose in. Remember, active transport means you got to push something against the gradient. Okay? So secondary active transport is regulated, okay, whereas primary active transport is constitutive, it's always on. Okay? And secondary can never be a uniport. Look at the definition. You need two substances traveling, right? That cannot be a uniport. Can secondary active transport be an antiport? Yes. Can it be a symport? Yes. All those modes exist. And when you do the kidney, you're going to see it. Okay. Any questions on this very heavy chapter? Okay. Cell transport 1. That's why I teach it hard in 102. But I haven't taught 102 in ages. So, and I don't know if they covered this detail in any 102. Does this make sense? So I have to cover it again. All right. Okay. 3-minute break. Okay, because I can see steam coming off through your ears. So 3-minute break, and then we'll start back during what? 2 words. I'm too tired for charades. Repolarization is a part of what?
Action
I'm getting a
potential.
heart attack right now. Action, but it's part of action potential, no? I'm done. How much did I scream about action potential? Girl, you should know this. You've had 9,000 years of me, and you're not answering. Next time I'm going to make you sit right here. I want to whack you if you don't answer. Repolarization is part of what?
Action potential.
I'm not hearing. Okay, I'm not giving up any more test questions until you all participate. Repolarization is a part of
Action potential.
Yeah, you'll talk when I give you a break and you have to gossip. Drama queens, answer me. It's part of action potential. Do you agree?
Yes.
So what did I just ask you? Does repolarization happen due to leak channels?
No.
No.
No. When are leak channels working? I'm here. When are leak channels work? Check your notes. It's not an exam. When are leak channels working? Huh? Are you listening to what I'm asking? When are leak channels working? Resting memory, wrong. Huh?
Aren't they always on?
Always on. They're always on. Are leak channels working during an action potential?
Yes.
Yes, but it's not significant. Am I making sense?
Yes.
Because voltage gated channels are open. Do you see how anal you have to be on these things? Hello?
Yes.
Yes.
That's right. Good morning. So leak channels are always working, just like what else is always working during an action potential? Sodium potassium pump. Exactly. Good. So they're both working constitutively. Are we clear?
Yes.
But it's not significant. So how does a leak channel contribute to action potential? It doesn't. Are we clear?
Yes.
Particularly potassium leak channels. Because when you open voltage gated channels, you're opening the floodgates. Who cares about leak channels? 2 ions coming in at a time versus 2,000 coming in at a time. Are we clear? It's going to be me and 4 people after this exam, isn't it? In the class. That's happened before. Anyway, so pay attention, people. If you didn't think about all this, time to think. It's still early. I'm so glad I did this review. Okay, my vocal cords aren't, but I'm so glad I did this. So what else was I going to ask? So during repolarization, voltage gated channels work. Are we clear?
Yes.
And they're more significant than leak channels. Way more significant than leak channels. If you didn't know this, write it down. So sodium versus potassium movement, right? So let's define these phases. So depolarization, you have 2, right? You have slow and you have fast. True or false? Depolarization can happen only due to sodium influx.
False.
False. What else can influx and cause depolarization? True, but that only happens in the year. Potassium will never influx, remember? Because potassium is high inside, low outside. Why would it come in?
Calcium.
Calcium, right? Not chloride, calcium. Chloride is negatively charged, right? And chloride doesn't move because its equilibrium potential is -70. Right? So in most cells it won't move. Okay? Now, what else can cause depolarization? I said this class number 1. When potassium is retained, so closure of leak channels can lead to depolarization. Are we clear?
Yes.
So 3 things, 4 things technically can cause this. Gain of sodium ions, gain of calcium ions, loss of negative ions, or retention of positive ions. Are we clear? Meaning you close leak channels. If I close sodium leak channels, what would happen?
Nothing.
If I were to close all the sodium leak channels in a cell,
Oh.
you will hyperpolarize slightly. Correct. It won't be a big, thank you. It won't be a big change, but you will hyperpolarize. Okay? You will go more negative from rest. Okay? Because that sodium leakage is what keeps the voltage at -70 and not -84. Are we clear?
Yeah.
Remember, that's what happens during restoration, right?
Now lost me on this. Na plus goes into the cell. If you're closing the leak channels, why doesn't the cell become more positive?
Because Na plus will not go into the cell. When Na plus goes into the cell, it has a big effect on positive charge, right? Sodium coming in will make the cell a lot more positive, right?
I have the Na plus K plus.
Mixed up, yeah.
Yes.
But very important question though. If sodium leak channels are closed, sodium will not enter. Okay? And remember, you have a predominance of negative charge, right?
Yes.
So you will become slightly more negative. But let's focus on that a little bit more. Why only slightly? Huh?
Sodium ions are highly restricted.
Yes, sodium ions are highly restricted. And the idea is you still have potassium inside the cell. Correct?
Yes.
And remember, if you close off the sodium leak channels, right, in general, it's going to be a very slight hyperpolarization, but it'll keep on decreasing. But it's going to happen very slowly. Are we clear? It's going to keep on decreasing because your sodium potassium pump is still pushing ions out. Are we clear?
Yes.
And sodium concentration will go to 0 and the pump will stop working. Remember this in PhysioX? If
Yes.
sodium concentration was 0, the pump stops. Good morning. The pump will
Yes.
stop. Okay? So when you choose not to eat salt, and you don't eat salt for several days, that's what happens. Your sodium potassium pump stops, and what did I tell you sodium potassium pump was? Your life. When it stops, it's bye-bye time. Are we clear?
Yes.
Okay. Okay, what is resting potential? So remember, equilibrium potential depends on the concentration of the ions, and it depends on the charge of the ion, right? So if you have a negatively charged ion, okay, that is very high on the outside of the cell, right? So let's say very high and it's on the outside of the cell. What will prevent it from coming in? If the cell became super negative, correct?
Yes.
It will repel that charge. It won't allow it to come in. Exact opposite situation. If you have phosphate, which is a highly negatively charged ion, but it's abundant inside the cell, what will prevent it from going out? If it's negative outside, or what inside?
Positive inside.
Positive inside, right? If it becomes extremely positive. So the equilibrium potential for phosphate will be a high positive number. Are we clear? Everybody got this?
Yes.
Think, don't memorize, think. Okay, very important. Because I can give you a very hypothetical situation here. Okay, voltage gated sodium channels. They have two parts. They have the M gate and the H gate. M gates flicker. H gates are a ball, right? M gates are called activation gates. H gates are called inactivation gates. Inactivated and closed are not the same thing. Inactivation means the H ball is trying to get out, and therefore it electrically and physically blocks ions from coming in. And remember, I posed this question for you, right? If H gates became negatively charged by mutation, right? Then you will not have action potential. Are we clear?
Yes.
Leak channels are still working. Sodium potassium ATPase is still working. But what will not work is the voltage gated sodium channels. And therefore, depolarization will not happen. Are we clear?
Yes.
Yeah.
So which depolarization will not happen? Slow depolarization to threshold. Fast depolarization from threshold to amplitude. Both. Neither. So
What's the
both
first
will not happen, or both will happen. If H gates were negatively charged, okay? Tell me, will M gates work?
Yes.
M gates will not work. M gates cannot work because think about it. If H gates were negatively charged, they're going to attract the sodium and bind to it, correct? What's going to separate them? Nothing. Guys, if you have two magnets that are attracting each other, don't you have to apply force to separate them?
Yes.
So if sodium is going to bind negatively charged H ball, what causes them to separate? Nothing. You've just poisoned the channel. Am I making sense? Sodium will just accumulate. Are we clear? So the channel stops working. Do you understand?
Yes.
So you will not have slow depolarization, you will not have fast depolarization, you will not have an action potential. Here's a tough question. Can hyperpolarization happen? Listen to my question carefully. Can hyperpolarization happen if you have this kind of a mutation? Define hyperpolarization. See, again, you're making that mistake. Going right to Jeopardy mode. Think of the definition. What's hyperpolarization? Becoming more negative from negative 70, correct? Yes?
Yes.
Can that happen if the H gate is stuck to the M gate? You're not thinking.
Yes.
Hyperpolarization can happen if voltage gated potassium channels open. If I got potassium channels to open, remember in graded potentials you can go hyperpolarize from rest. Yes?
Yes.
So the answer is yes, it can work. Now here's the harder question. Will repolarization happen? What's the definition of repolarization? You're going from amplitude to rest, yes? How do you get to amplitude? Voltage gated sodium channels. If voltage-gated sodium channels did not work, can you get to amplitude?
No.
Which means can repolarization happen?
No.
No. But hyperpolarization can. Are we clear? Hyperpolarization is independent of depolarization. Okay? Definitely it can happen. Please think about that. Very, very important. And the fact that you have inhibitory neurotransmitters, the fact that graded potentials can be negative, right? They're all evidence that hyperpolarization happens without a preceding depolarization. Are we clear?
Yes.
Okay. So closed versus inactivation, I talked about that. Closed is when the H ball is in the cytoplasm, but the M gate is solid, it's not flickering. Okay? Inactivation is when the M gates are flickering at their maximum, but the H gate is blocking everything. That flickering has no consequence. Are we clear?
Yes.
Okay. Absolute and relative refractory periods. Refractory means you cannot generate a new action potential cycle. Here's another example of not knowing definitions properly. When I say action potential, I need you to know that it's a cycle. Everybody say this, action potential is a cycle.
Action potentials are cycles.
It's not everyone.
Action
I'm going to
potential.
sit here and wait till everybody screams it at me, or we're not moving forward.
Action
Action
potential.
potentials
Action
are.
potential is a cycle, right? So it starts from rest and goes eventually back to rest. Are we clear?
Yes.
Okay. So absolute refractory period is when you cannot generate a new action potential cycle. Absolutely not. Are we clear?
Yes.
That's because either the sodium channels are at Tmax, voltage-gated sodium channels, not leak channels, so voltage-gated sodium channels are at Tmax, or they're inactivated. Are we clear?
Yes.
So you need to know your definitions, okay? Those constitute the absolute refractory period. What's relative refractory period? Where a second action potential cycle is possible,
But difficult.
but the cell is still leaking positive ions, right? Which means you're going through hyperpolarization or restoration. So it's either that top triangle or that bottom triangle. Make sense?
Yes.
Relative refractory period simply means that it's harder and harder to generate a second action potential when you're hyperpolarizing. Are we clear?
Yes.
Okay. I talked about graded potentials. We talked a lot about graded potentials. Graded potentials do not use voltage-gated channels. And I wrote this all out. It's all in your notes. Okay? Graded potentials do not. Guys, if you sit down and copy these 10 pages of notes, it's going to take you 20 minutes tops. Your learning will go up 1,000-fold. Literally, take my notes today, sit down. You don't have to be mentally alert at all for this, right? Take out another piece of paper or take out your iPad, open a new set of notes, and just recopy the whole thing. Yes?
Yes.
Yes.
You will be amazed at how much you've learned and how quick. Okay? And again, I promise to try to add questions to your practice exam. The practice exam was not meant to include neurons, okay? Last time we didn't do neurons. I gave them an exam right after action potentials. But it's still helpful, okay? And I'll try and add questions to it, okay? It's not a promise, but I'll try, okay? Maybe I can just add my 103A old exam questions to it. It's something better than nothing, okay? They're all screaming about it anyway, so it should work out for you. That's what I'll do. Okay, so action potentials are all or none, right? They're all or none, whereas graded potentials are a wide variety, right? Yes?
Yes.
And they can be depolarizing or hyperpolarizing. And graded potentials reflect the stimulus strength, and graded potentials often occur on cell bodies of neurons. Are we clear?
Yes.
Action potentials occur along the axons. A for action potential, A for all or none, A for axons. Okay? So neurons have a cell body. They have an axon. An axon can be myelinated, which means saltatory conduction, right? Signals leap from one node to another. which is fast or slow?
Fast.
Fast. Remember type A fibers, right? In the PhysioX experiment. Remember, the larger the diameter of the neuron, the faster the signal moves. This is an important relationship. Okay, diameter. The speed is directly proportional to the diameter, meaning the greater the diameter, the faster the signal moves. Okay, and signal here is action potentials, right? We're talking action potentials. Glia, these are additional cells. Glia are not on the exam. You don't need to know those. That's anatomy, okay? I just included it here. It's not on your exam. I'm not going to include glia on the exam. Too easy. Except one thing I want to ask you. Astrocytes have to coordinate nutrition and reactions and processes amongst each other, right? What will you find among astrocytes? If you have to coordinate and synchronize gap junctions, astrocytes are rich in gap junctions. Okay, good. Central nervous system, brain and spinal cord. Peripheral nervous system, you have sensory neurons, right, which could be unipolar, bipolar, or multipolar. Okay, or motor neurons, which are primarily multipolar. Okay, motor neurons are primarily multipolar. Okay, motor, multi, MM, right? So make that relationship. Sensory division sends signals to the brain or the spinal cord. Motor division brings signals from the brain or the spinal cord to effector organs. Somatic is a motor division exclusive to skeletal muscle. So somatic deals with voluntary movement. Autonomic deals with everything else, right? It's autonomic, right? Sympathetic, parasympathetic. I talked about neuron types already. Unipolar and bipolar. These neurons divide a lot, so you get cancer of these neurons a lot. They divide a lot. Multipolar neurons are usually very, very terminally differentiated. They do not divide for the most part, with the exception of hippocampus. They don't divide. Multipolar neurons do not divide. Bipolar neurons for sure do. Unipolar, I'm not sure. Okay, but I know bipolar neurons do because of nasopharyngeal carcinoma and retinoblastoma. Right? These kinds of cancers, they're cancers of neurons in the eye and the nose. Okay.
Yes.
So let's see, where are we? Sympathetic is fight or flight or fright. Parasympathetic is rest and digest. Okay. Dendrites are the receiving portions, right? Graded potentials happen at the dendrites. By the way, where does summation happen? Cell hillock, right? Axon hillock is where summation happens. Are we clear?
Yes.
The dendrites and the rest of the cell body are all receptive, meaning they have receptors for neurotransmitters. Thank you. They have receptors for neurotransmitters. By the way, here's something that I sent by announcement. If you're not getting my announcements, you're going to miss out on this, on extra material which you're responsible for. So I'm going to give people benefit of doubt who didn't read my. You should make your announcements come to your email. Are we clear?
Yes.
If you haven't done that, you should have done that 10 days ago. Okay? So, what did I send through announcements? And tell me if you didn't get that announcement. A given neurotransmitter can have contradictory effects if its receptors are different. Are we clear?
Yes.
When we start the heart, all the way to the end of the semester, you're going to be seeing this over and over and over again. Are we clear?
Yes.
Okay, so in some blood vessels, okay, sympathetic will vasodilate. In some blood vessels, it will vasoconstrict. Okay, so if you have alpha 1 receptors, you will vasoconstrict. If you have beta 2 receptors, you will vasodilate. Do you understand what I'm saying?
Yes.
So depending on which receptor you have a completely opposite response. When you dilate, you relax smooth muscle. When you constrict, you contract smooth muscle. Do you see how they're opposites?
Yes.
Okay, so very, very important, and you're going to see how that works next week. when we do muscle contraction. Okay? Watch the muscle contraction video if you can, okay, before you come to class next week. So axon hillock is where summation happens. You have simultaneous neurotransmitter release. That's going to be what kind of summation?
Spatial.
Spatial. S for simultaneous, S for spatial, right? So what is spatial? You have multiple different kinds of neurotransmitters. And there was a brilliant question that a 103A student asked me last semester. What if you had two opposite receptor types? Yes, think about this question, really important question, because I didn't know the answer. I had to go research into this, and it took me a month to figure out the answer to this. Maybe I should be careful in admitting to these things, but we don't have that person here, so I'm okay. So it took me a month to find the answer, because this isn't easy. So you have two neurotransmitter receptors that are opposite to each other, right?
Mm-hmm.
One will activate contraction of smooth muscle, one will, let's say alpha 1 and beta 2, right?
Mm-hmm.
Those are adrenergic receptors. Adre means adrenaline, right? Or epinephrine, which is what nervous system? Sympathetic, correct? So if you have both of those and you're simultaneously releasing norepinephrine, yes, what will happen to a cell? Well, if you're releasing them in equal amounts, and if the receptors are in equal amounts, that's dumb. You've just wasted a lot of energy. Because you're going to be using ATP to make cyclic AMP. You've just wasted energy, right? So that will never, ever happen. Okay, that's why it took me a month to figure this out. Because there is never a cell type where both of them are expressed right next to each other. So you cannot have spatial summation with the same neurotransmitter. Do you see that? What is the point of that? One's going to be plus, one's going to be minus. They're going to cancel each other out. So all the energy you spent releasing the neurotransmitter, because you use bulk transport needs energy, and all the energy you need to pump that calcium back out, you've just wasted all of that. Do you understand what I'm saying?
Yes.
One big waste, right? So spatial summation will only happen with multiple neurotransmitter types. Do you understand that?
Yes.
Otherwise, it's physiologically irrelevant. Okay, so keep that in mind. Spatial includes hyperpolarization, right? Whereas temporal can only be depolarizing. Are we clear?
Yes.
There is no negative. There's only on or off, and that's it. There's no going negative. Are we clear? Okay. So excitatory neurotransmitters will depolarize only at the postsynaptic membrane. Inhibitory will hyperpolarize at the postsynaptic membrane. You understand what postsynaptic means, right?
Yes.
True or false? Preganglionic and postganglionic are one type. Listen to my question carefully. Preganglionic and postganglionic are one type of presynaptic and postsynaptic. If you don't understand this, write this down. Preganglionic and postganglionic are one example of presynaptic and postsynaptic. What's a synapse? The gap between two neurons, correct? In a ganglion, do you have synapses? Again, I repeat, a synapse is when two neurons meet. Do two neurons meet in a ganglion? Yes. Correct? So yes, it is an example, absolutely. Okay? Of course it's an example. Okay, so again, when you do terms, think like this. It's important. It helps you concrete the definitions. Okay? So, okay, moving on to brain, the cerebral organizing principles. This is on your exam. All of this is on your exam. I wrote the sheet from your exam, actually. So, with very few exceptions. Almost all of this is on there. So, cerebral organizing principles. Unequal areas. Unequal areas. Lateralization. What's lateralization? Well, first of all, what's unequal areas? The more you use something, the bigger the region in the brain, correct?
Yes.
And remember, you have sensory and motor. Motor and sensory. Sensory is in the back, motor is in the front. So, if you can change these areas, what is that property called? Plasticity, thank you. It's called plasticity, if you can change these areas. Okay, good. And contralaterality, what is that?
Controls the opposite.
One hemisphere controlling the opposite hemisphere, correct?
Yes.
And lateralization means the uniqueness of the hemispheres, right? For example, Broca's area is only on the left hemisphere, not on the right. The hemispheres aren't identical functionally. Do you see that?
Yes.
Right? They're overlapping, but they're not identical. Okay? So ascending means going to the brain. Descending means coming down from the brain, right? Ascending are sensory tracts. Sensory tracts are ascending. What tracts are descending?
Motor tracks.
Motor tracts are descending. Okay, good. Nuclei versus ganglia. Nuclei and ganglia are groups of neurons, right?
Mm-hmm.
Nuclei is if it's inside the CNS. Ganglia if they're outside the CNS. Everybody get that? So in the peripheral nervous system, they're called ganglia. In the central nervous system, they're called nuclei. Okay? Now, where are nuclei important? Your subcortical areas. Remember? Subcortical areas are arranged as nuclei. Your thalamus is arranged as nuclei. Hypothalamus arranged as nuclei, right? Those are all nuclei. And then pons and medulla oblongata, they're also nuclei. They're all nuclear organization, clear? Because they do many different functions. Each nucleus has a unique function. Are we clear?
Yes.
Okay. Cholinergic receptors, these are acetylcholine receptors. Adrenergic would be adrenaline, right? Cholinergic means acetylcholine, right?
Yes.
So cholinergic receptors can be nicotinic. These are direct acting receptors. Direct acting because they are themselves ion channels. Are we clear? Example, skeletal muscle. Muscarinic are indirect acting. Okay, muscarinic are indirect acting receptors, but they also respond to acetylcholine. Lastly, sympathetic nervous system is thoracolumbar, and they're arranged as chain ganglia. They're connected to each other. Why? Sympathetic ganglia are connected to each other. Two things you need to know. They're connected to each other. Why? And they're found right next to the spinal cord. Why? Check your notes. First of all, if your thoracolumbar outflow, do you have a cognition part here? Let me ask you this. Does fear have to be rational?
No.
In fact, most of the time it's not. Right? You step on something, it's wet and feels like a rope. You get the bejesus scared out of you because you think it's a snake. Make sense?
Yes.
Your body immediately responds. There's no waiting for you to think. You don't have time to think. Do you understand? Okay? If you see smoke in the room, but it's smoke because I just added sodium hydroxide to bicarbonate, so you get smoke, that's not what you think. You walk into the room and it's smoking, you're going to turn on the fire alarm. You're not going to stand there and go, I want to find out why it's smoking. Nobody has time for that, okay?
Yes.
But what about craniosacral or cranial sacral? That would be which system? Parasympathetic, because you need to think, right? Parasympathetic requires thinking. Even for erection, you need thinking, correct?
Yes.
Hello?
Yes.
I see this thing. I'm turned on by this. You need your brain to work for that. Okay? Yeah, as far as I know, humans, again, are the only exception here because humans can get an erection during a coma. You can mechanically stimulate the clitoris as well as the penis. But even in species like wolves, where the clitoris is 10 times longer than the penis. Okay? The clitoris is much longer. The female is the one who chooses the male, and the male stays home and takes care of the pups. The female hunts. The male's a homemaker, okay, and it's affectionate, and his penis is teeny weeny, okay? The female has the longer clitoris, okay? So these things exist, okay? What is my point? The point is, erection is important in the female also, so don't ignore it, okay? So craniosacral is primarily for two purposes. One is for peeing, okay, and two is for erection. Are we clear? Okay. All the other ganglion in parasympathetic are found near the target organ, right? So parasympathetic has long preganglionic and short postganglionic, and they both release acetylcholine. Reviewed what kind of receptors used in each case. Remember, at the ganglia, it's always nicotinic. Are we clear?
Mhm.
And at the target, it's always muscarinic. Okay? So slow. And always think about the heart. You don't want to do anything fast with the heart. Are we clear? Or anything rapid. It has to be gradual. Okay. Any other questions? Anything I didn't really do here? I don't think so. And this completes 15% of the course. 1-5. Feels like 50, doesn't it? But it's only 15. From now till maybe, I would say April, there's going to be a lot of memorization, but not a lot of depth. Make sense? There's going to be memorizing quite a bit, but not a lot of depth. And we're going to do lots and lots of action potentials, which you've already learned. So it's not going to be so bad. Okay? All right, class dismissed. I will see you all Tuesday. We will start muscle, okay, during lecture. Okay? So when does the exam start, everybody? Lab day, what time? 11:15. Exam starts 11:15. And what are you going to do as soon as you go home today, if you haven't done it already? Announcements. Make sure you get them in your inbox. And check your Ohlone email, because all my emails go there. Okay? All right. Have a wonderful weekend. I will see you all next week. And when are my office hours? 9. On Zoom. Don't come to campus. I have to say that too.